it's understanding how the host immune system responds to the virus and defining that protective immunity. We're just now learning about how people who have been infected might be protected from reinfection. We've heard a bit about the convalescent plasma that's being used in some patients. This transfers antibodies from someone who's recovered from the infection into someone who's actively infected as a way to help the infected person clear that infection. That's been showing some promise. So there's some evidence that antibodies might be playing a role in helping to clear the infection, which would then make that a likely target of vaccine-induced immunity. But we still need to understand what antibodies, what sort of level of antibodies we want, and how the virus will respond to interaction with those antibodies that will define, the development of a vaccine. My lab at the University of South Dakota studies viruses and vaccine development in particular. We study influenza viruses in the context of vaccine-induced immunities and secondary bacterial infections. And with this new coronavirus pandemic, we are getting involved in learning a little more about the immune response against SARS-CoV-2, trying to help in the development of vaccines against this new virus, using our knowledge of how the immune system interacts with influenza to then try and develop vaccines that would be useful against SARS-CoV-2. This virus is an RNA virus, similar, influenza is also an RNA virus. RNA viruses have a much higher mutation rate than, say, a DNA virus. And we already know that it does mutate. And you can tell that by looking at the genetic code. Now, the important mutations for viruses are going to be the ones that change the ability to transmit from person to person or between species, the sort of severity of the illness that it causes, and then the ability to escape the host immune system. And those are the questions that we don't know about this virus. And we'll have to learn as we, again, gain more experience with this antibody-mediated immunity, how the host is protected, and then the development of vaccines. And so those are going to be the sort of mutations we're going to have to keep an eye out for. With influenza, as I mentioned, the vaccines were developed and released in 1945. We didn't really know influenza changed in the way that it does until 1947, when the vaccine, while it induced a strong immune response, didn't protect against the virus that was circulating. And that's when they realized this virus changes, and then they established a surveillance network in 1952 to then start tracking those changes and coming up with the system that we have today, where we change the vaccine based on those changes.